A very good morning to you all. In our previous session, we have discussed in detail about the different parts of the fermenter and the function associated with each part of the fermenter. And we were discussing about the fundamental steps in recovery and purification of the fermentation product. Then we also discussed about the unit operations. That is, each unit operation is based on exploiting unique physico-chemical properties of the product. In today's session, we will further discuss on various purification methods adopted during fermentation process. So essentially we are discussing in detail about the recovery and purification of fermentation product in the process of fermentation. Now the question is whether each operational unit, as discussed in the previous session, is 100% efficient or not, be it distillation, precipitation, fractionation, evaporation. So whether each unit operation is 100% efficient or not. It cannot be 100% efficient for the reason each unit operation or each step in the process of purification or in the downstream processing, it involves the loss of some quantity of the product. Hence, it will not be 100% efficient, and added to this, the product degradation may have occurred. So even if the percentage recovery for each step is very high in the sense it will not reach the scale of 100%, say for example 90%. And in the subsequent steps of purification or the recovery and purification, say for example some 4 to 5 steps down the lane, you may end up with only 60% of the initial product. So what you observe over here is that there is a loss of some quantity of the product as each operation is not 100% efficient. So the more number of steps in the recovery and purification process, more loss of the product will be there. So it is important that the minimum number of unit operations are designed to maximize the product recovery. Right? So if there are minimum number of unit operations, then there is maximum product recovery. If you increase the number of steps or unit operations, then you end up with losing the product, some quantity of product, as I said, maybe from 90% to 60%. So on an average 30% of the product is lost. So it is always recommended to keep the number of steps to the minimum possible so that you end up with a good quantity of the product. Now, what should be the choice of recovery process? What are the basic criteria in choosing the recovery process? There are 8 different criteria I have listed over here, which are very much important for choosing the recovery process. Number 1, the intracellular or extracellular location of the product. So, when you are choosing a recovery process, the first criteria you should concentrate more is whether the product you are trying to recover and purify, is it intracellular or extracellular, whether the location of the product is inside the cell or outside the cell. Second criteria, the concentration of the product in the fermentation broth. What is the concentration that is available, the approximate concentration that is available in the fermentation medium. The third criteria, the physical and chemical properties of the desired product. So that if you know the physical property and as well as the chemical property of the product you are trying to recover and purify, then it will be much helpful to choose an appropriate separation procedure. Next criteria, the intended use of the product. What is the specific application of the product? Number 5, the minimal acceptable standard of purity. So, you should be able to reach the minimal acceptable standard of purity. So, whatever the purity standards that is set, so you should be able to reach that minimum standards. Number 6, the magnitude of biohazard of the product or the broth, whether it is biohazardous. If it is so, what is the magnitude of biohazard nature of the product or maybe even the broth. Number 7, the impurities in the fermenter broth. What are the impurities? What are the possible impurities that are present within the broth? Number 8, the marketable price for the product. Right. So, these are very, very important criteria to be understood, to be thoroughly worked out before choosing the recovery process. That is, the intracellular or extracellular location of the product, the concentration of the product in the fermentation broth, the physical and chemical properties of the desired product, the intended use of the product, the minimal acceptable standard of purity of the product, the magnitude of biohazard of the product or maybe the fermentation broth, the impurities in the fermentation broth, and the marketable price for the product. Okay. So, this figure gives you an idea about the stages in the recovery of product from a harvested fermentation broth. So, it is a generic illustration of various stages in the recovery of product from a harvested fermentation broth. So, to start with, you have the fermentation broth, then you remove the solids, then the cell disruption occurs, primary isolation of the product, purification and concentration, and finally you end up with the isolation of the final product. It is a generic flow chart of various stages in the recovery of the product. So, the main objective of the first stage for the recovery of an extracellular product is the removal of the large solid particles and microbial cells. Okay. So, assume that you have your product which is in which the location of the product is extracellular. So, the main objective of the first stage for the recovery of an extracellular product is the removal of the large solid particles and microbial cells. So, this is done by usually the process of centrifugation or by making use of filtration. In the next stage, the broth is fractionated or extracted into major fractions. So, that is done by using ultrafiltration, reverse osmosis, adsorption, or even the chromatographic techniques, say for example ion exchange chromatography, gel filtration chromatography, or affinity chromatography. Then we have liquid extraction, two-phase aqueous extraction, supercritical fluid extraction, or precipitation. Okay. So, in the next stage, the broth is fractionated into major fractions, and this is achieved by using ultrafiltration, reverse osmosis, adsorption or ion exchange chromatography, gel filtration, affinity chromatography. Then we have liquid extraction, supercritical fluid extraction, precipitation, two-phase aqueous extraction. So, there are various stages wherein you can adopt various recovery process. So, which recovery process has to be adapted is dependent on certain criteria which we have discussed in detail in the last slide. So, afterwards the product containing fraction. So, after you fractionate the broth, so the product containing fraction is purified using fractional precipitation, and further you can purify using precise chromatographic techniques, and then crystallization of the product happens. So, in the crystallization process, it is like the product is crystallized and it is highly concentrated and it is essentially free from impurities. So, you would have achieved the minimal standards of purity of the product. Right. So, it may be possible to modify the handling characteristics of the broth so that it can be handled faster with simpler equipment, making use of number of techniques. Right. So, handling characteristics of the broth with number of simpler techniques to obtain the product in the desired quantity is an important concept in the recovery and purification process. So, what are those simpler equipment which makes use of number of techniques to obtain the product? Number 1, selection of a microorganism which does not produce pigments or undesirable metabolites. So, the first and foremost factor to be remembered is in the choice of the microorganism or the biological source. The selection of the microorganisms should be in such a way that it does not produce any pigments, because you may not be interested in the pigment production, or undesirable metabolites. You should choose a microorganism or a cell population wherein you can expect a large quantity of the product is produced with no pigment production or undesirable metabolites. Then modification of the fermentation conditions to reduce the production of undesirable metabolites. Even if the microorganisms tend to produce undesirable metabolites, and microorganisms produce these metabolites, and the factors which influence the process of production of metabolites could be the pH of the medium, the temperature, the available nutrient source. So, if you can modify the fermentation condition itself to reduce the production of undesirable metabolites, so that influences on maximizing the yield of the product and easy recovery of the product. Third one, precise timing of harvesting. So, you should be well aware of the time of harvesting the product. Number 4, pH control after harvesting. So, this is very, very important, otherwise degradation of the product may happen. So, pH control after harvesting. See, these are some of the simpler equipment or the technique that maximizes the yield of the product, that is selection of microorganisms which does not produce pigments or undesirable metabolites, modification of the fermentation conditions to reduce the production of undesirable metabolites, precise time of harvesting, pH control after harvesting. Number 5, temperature treatment after harvesting, which is very, very important, because the temperature within the vessel may not be matching the temperature outside the vessel. So, temperature treatment after harvesting is equally important. Number 6, addition of flocculating agents. Number 7, use of enzymes to attack cell walls if it is intracellular, so that the product comes out of the cell. Right. So, it must be remembered that the fermentation and the product recovery, they are the integral parts of the overall process. They are not separate entities. Fermentation and recovery of the product are interdependent. They are the integral parts of the overall fermentation process. So, because of the interaction between the two phases, the fermentation and the product recovery phase, neither phase should be developed independently. So, when you are designing a process, it means you are designing a fermentation process as well you are designing a product recovery steps, so that you end up with maximum yield of the product with good quality. So, whenever you are designing a process, because there is an interaction between the fermentation process and the product recovery stages, neither stage should be developed independently. Is that clear? So, how to remove the microbial cell and other solid matters? The microbial cells and other insoluble materials, they are normally separated from the harvested broth by filtration or centrifugation, as I discussed in the previous slide. So, they make use of filtration or centrifugation. Because of the small size of the microbial cells, it will be necessary to consider the use of the filter aids, and you can have the heat and flocculation treatments along with filtration or centrifugation, so that you increase the sedimentation rates in centrifugation. And flocculation can also be used in the downstream process to help the recovery of the product. So, in order to remove the microbial cells or any other solid material, the process makes use of filtration or centrifugation or flocculation, right. Then we have foam separation or flotation. Foam separation or flotation depends on using the methods which exploit the differences in surface activity of the material. Right. So, the material may be whole cell or may be the molecule. You can have two types. It can be the whole cell or may be the molecule. So, the molecule may be in the form of a protein or a colloid. Now, this is selectively adsorbed or attached to the surface of the gas bubbles or the foams, and these gas bubble rise through a liquid, that is the medium, fermentation medium, and finally that has to be concentrated and then the product is removed using the skimming. Right. So, the material may be the whole cells or the molecules such as the protein or colloids. So, these they get adsorbed or attached to the surface of the foam or the gas bubbles, and they rise through the fermentation medium, which is supposed to be concentrated or separated, and then finally they are removed by skimming. Right. So, this is a beautiful illustration which gives you an idea about the foam fractionation. So, we have something called as sparger over here. So, we have seen the function of the sparger in the last session under different parts of the fermenter and associated functions. Sparger function is to introduce sterile air or oxygen into the fermentation medium. Then what happens? There is foam formation, in which the whole cell or the molecule, maybe protein in the form of protein or colloid, they bind and they overflow, and then we have foam breaker, and then the product recovery is continued. Right. So, it is or it may be possible to make some materials surface active, because foam flotation makes use of the property of the surface active of the materials. So, you can make some material surface active by applying the surfactants, and these surfactants could be the long chain fatty acids, amines and quaternary ammonium compounds. Right. So, the materials made surface active and collected, they are called as colligents. So, what are colligents? Colligents are the materials whose surface active is made by applying the surfactants such as long chain fatty acids, amines and quaternary ammonium compounds. And here the surfactants are called as collectors. So, the materials made surface active and collected are termed colligents, whereas the surfactants are termed collectors. Right. So, when developing this method of separation, the important variable which may need experimental investigations are: number 1, the pH, the airflow rates, the concentration of the surfactants and colligent collector ratio. So, here the colligent, as I said, is the material whose surface active is made by applying the surfactants, and surfactants are termed as collectors. So, what is the ratio of colligent to collector, and what is the pH level, what is the airflow rate, what is the concentration of the surfactant. So, all these variables are important and they need experimental investigations when you are using foam flotation process for recovery of the product. Right. Next we have precipitation. It is another unit operation process. So, the precipitation need not always be conducted at a particular stage of fermentation process. So, you can conduct the precipitation at various stages of the recovery. Need not be always in the initial phase or in the final stage, not like that. You can use the precipitation as unit operation in various stages of the product recovery process. So, it is very much important and useful because it allows enrichment and concentration in one single step. Thus, it saves the volume of the material required for purification. Right. So, it is possible to obtain some products directly from the broth by using the precipitation. You need not have an intermediate purification step. So, you can directly obtain some of the important products just by employing the precipitation method. Right. So, the precipitation as an unit operation in the fermentation process can be conducted at various stages of the product recovery process. It is useful, very much useful, because it allows enrichment and concentration in one single step. And in some cases, some of the products, or maybe even to remove some of the impurities directly from the broth, then precipitation is the best choice. Right. So, what are the typical agents used in precipitation which render the compound or the analyte or the product of interest insoluble? So, precipitation is essentially making the product or the compound or the analyte insoluble and then removing it from the solution. So, this is the basic principle. So, precipitation means you are trying to remove the product of interest out of the solution. So, what are the typical agents that can be used in the process of precipitation to render the compound or analyte or the product of interest insoluble? Number 1, acids and bases. Number 2, we have salts such as ammonium and sodium sulfate. Number 3, organic solvents. Number 4, non-ionic polymers such as polyethylene glycol. Number 5, polyelectrolytes can also be used. Number 6, protein binding dyes, as an example the triazine dyes. Number 7, affinity precipitants. Number 8, heat treatment as a selective precipitation and purification step. Heat treatment can be or it is applicable only for the thermostable products. So, moving back, we have 8 different types or methods to be used in the precipitation process to make the analyte or the compound or the product of interest insoluble. Got it? That is number 1, the acids and bases. So, to change the pH of the solution until the isoelectric point of the compound is reached, and the pH equals Pi, then there will be no overall charge on the molecule and its solubility is decreased. When there is no overall charge on the molecule, so you can do this by changing the pH of the solution until the isoelectric point of the compound is reached. When this happens, the solubility of the compound or the product or the analyte is decreased. So it gets precipitated. Is that clear? So I'll stop over here my today's session. We can continue our discussion in the next class. Thank you very much.